Hello friends, welcome to back my channel. Hope you all are doing well. So today I will discuss about TB Platinum, what is the principle of TB Platinum, assay procedure and interpretation of the results. So what is a TB Platinum? TB Platinum is based on the principle of IGRA, interferon gamma release assay. So it is a quantitative and check for TB infection and TB disease. So in that we have microvels, standard HRP conjugate, sample dilution, standard buffer and wash buffer, chromosome solution A, chromosome solution B and stop solution. So let's get it started. So what is the things we have in that kit? First one is microvels and N and T background control tubes, and this is one wash concentration for washing purpose, and second one is sample dilution, and third one is standard dilutions, and fourth one is standard. So standard, it is for this standard is used for kit validation when we have received new lot. So every lot we have to process standards also. So this one is for chromosomal solutions. This A and B solutions we are using, and last one is stop solution. So this reagent for ELISA, we are using this all reagent for ELISA. This is the TB Platinum is based on ELISA technique, and in that kit we have testing culture tube, this called T tube, and second one is background control culture tube, is called N tube. So testing culture tube, if patient have TB infection, so in that absorbance is high detected, so it means TB is positive, and absorbance is less than 14, it means TB is negative. So next step, we have diluted standard and reconstitute the standards. So first we have to take wells. So how many amount of sample we have, and 1 to 6, like 400 to 12.5 we have concentration. So according to this concentration we have to dilute the standards, and how many amount of sample, first we have to count, and then according to the sample break the well and set it in the microplate. So we have 6 standard and 4 sample. So for 6 standard, 6 well we have taken and 4 samples. So standard, this one is standard, 1.2 ml deionized water, and reconstitute this standards. So add distilled or deionized water into ampule according to the volume, and indicate on the label of ampule, and reconstitute the freeze dried standard after 2 to 3 minutes. So the standard is completely dissolved, gently mix and until it is homogeneous. So safely you have to break this ampule and then reconstitute with distilled water or deionized water. So 1.2 means 1200 microliter. So if you don't have 2000 microliter pipette, so take 1000 microliter pipette. First you have to take 1000 microliter, and second step you have to take 200 microliter. So total amount is 1200. So 1200 means 1.2 ml. So 1.2 ml you have to reconstitute in standard. So after reconstitution done, so our final concentration of this standard ampule is 400 pg/ml. So final concentration properly mix, and this last final concentration we have 400 micro pg/ml. So 400 pg/ml standard is prepared. After that, taken 6 well, like 6 tubes. First tube 200 microliter, uh, transfer 200 microliter in first tube. For standard ampule, 400 concentration, so we have to take 200 microliter concentration, and then next step, then second to six, take standard dilution buffer. Second to 6, take standard dilution buffer. So sample dilution is different or standard dilution is different. So for standard only we are using standard dilution buffer. So like 200, 100, then 50, 25 and 12.5 we have to take concentration of this standard. So first one 100 microliter, second to 6 standard dilution buffer we have to add it, and then serially diluted the sample, like 100 microliter, 200 microliter concentration to transfer 100, and then this concentration is final 200, 400, and then 200 to 100, 100 to 50, 50 to 25, 25 to 12.5. So the final concentration is our 400, 200, 100, 50, 25 and 12.5. Okay, then next in microwells, first take 50 microliter standard 1 to 6. So first 400 concentration take 50, then 200 concentration take 50, 100 concentration take 50, then 50, 25 and 12.5 same to take 50, 1 to 6 standard. And you have if you have 2 sample, 1 sample or 4 sample, take as per samples N and T control wells, and transfer first one 20 microliter, 20 microliter sample dilution buffer. So first we have to check if IDs is different for T and N tube, so correct this. And labeling is very important thing for TB testing, so please done correct labeling in TB vials or patient serum. So sample collection is done in heparin vial, and at 37 degree to 24 hours we have to give incubation N or T vials. So first in patient sample, take sample dilution buffer first and second well, and we have 4 samples, so according to that we have to transfer sample dilution buffer in only patient sample wells. So first, second, third and fourth. So for collection purpose, first things we have to take heparin and max 3 ml sample, sufficient sample for 3 ml, and then incubate. The first one is transfer for N and T, and then incubate 37 for 18 to 24 hours. So And first one, take background N tube 50 microliter sample, and second T for testing culture tube 50 microsample. So total for standard 50, for patient sample 20 plus 50. 20 means sample dilution or 50 means standard dilution. Okay, so after that, this is a protocol of first step. So next step, we have to add conjugate. So what is a conjugate? So what is a conjugate? So conjugate is a prepared horse radish peroxide conjugate anti interferon gamma antibody. So after done procedure, take 1 hours of incubation and keep in 37° in incubator. So after incubation, 1 hour incubation is done, so add 50 microliter HRP conjugate reagents. Yes, this one is HRP conjugate reagents, and then cover the plate and incubate 60 minutes at 37°. So safely cover the plate and incubate. So add directly in sample or standard dilution. After 1 hour of incubation, pour 50 microliter HRP conjugate in all vial. So standard also and sample also. So next step, again keep in 37° for 60 minutes and incubate in incubator. Incubate 37° for 60 minutes in incubators. So note down the time. So write down in label in that label. So this is very important things. Time is note down as per protocol. So after 60 minutes is completed, open and wash 5 cycle with wash buffer. So first take tissue rolls, and in washing purpose we need tissues for blotting. So dispense washing buffer, 20x concentration of washing buffer. So if 20x concentration of a washing buffer, how to dilute it? So if 20x concentration, so you have to need diluted. So 20x means 1:20. So take 1 concentration buffer and 19 part of distilled water. So blot first to 5 cycle. So wash 5 cycle with wash buffer. Then after 5 done 5 cycles of washing, so added 50 microliter A plus 50 microliter B substrate. So 50 microliter A or 50 microliter B substrate. So substrate is like color development. If color is not development, so it means our process is not working. So dispense 50 microliter chromosome A and 50 microliter chromosome B solution in each well, including the blank also, and tapping the plate gently and incubate plate 37 for 15 minutes. And important things, avoiding light. The enzymatic reaction between two chromosome A and B solution and HRP conjugate process produces blue color in the standard well and in the interferon gamma positive sample well. It means if TB is present the patient, so it color is formation like blue color. So standard well is if interferon gamma interferon is positive, it means its produce blue color. And then completions after completed 50 minutes, remove cover and added 50 microliter stop solution and take OD. So we have Merrill ELISA reader. So put this tray and operation you have. Afterward I will discuss in next video how to operate Merrill. So run and TB Platinum, select the test name, then select standard. So first to 6 standard we have to select. First standard 1, 2, standard 1, 2, 3, 4 and 5 and 6. So then take sample. So sample for one sample we have taken 2, so N and T background cell control, and so 4 it means 8 well we have taken. So run and wait for 1 minute, and then run is completed and validation of result. If completed, it passed, and if failed, so it display it is not passed, so it is printing, so it means it is passed. So then this is our absorbance values, like 12.5 is first concentration, 25, 50, 100, 200, 400 concentration. And then how we take calibration graph. So first we have to go calibration, then select the test TB Platinum, and after that if test is validated with date is coming, like we have process today, so date is coming like 5/7/23, active calibration. So plot, select and plot the graph. So this one is our standard ODs. So our standard ODs is first 6 to 1. So first is 12.5, second is 50, then 100, then 200, then 400. So this is our calibration uh, printout, and this one is how software IGRA software, Immunocheck TB Platinum. So we have to put ODs inside this side, OD side, and concentration of this we have to put. So our ODs is coming like that. So it means R2 is 0.987. It means our graph is valid. So after validated of the graph, after it means our kit is working properly. So we have to take patient sample